Hi, everyone. My name is Melanie Dizon, and I'm the Director of Education for the Davis Phinney Foundation. Today, I'm here with Pete Schmidt. Pete is a member of our board of directors at the foundation, is an advisor to our science advisory board, and is a longtime friend of the foundation. Today, Pete is going to be talking about a study he worked on about how infection with the virus that causes COVID-19 enhances susceptibility to oxidative stress-induced Parkinsonism.
First, I'm going to start with a quick primer on what is Parkinson's disease and how I think about Parkinson's when I think about it. So we're going to start going in, get a little insight into the top level issue. And the first thing I want to highlight is that the brain is a network. We think about brain cells, the neurons of the brain, and we think about how they are the components that make the brain, but actual thought happens at the synapse. That's where the signal crosses that little tiny gap between the axon and the dendrite of two connected neurons is the place where thinking happens. So we have a presynaptic and a postsynaptic cell. They pass a message from one to the other. And the cell body itself, which we always think of as so important, where the DNA resides in a neuron, it's really the battery. It's the thing that provides the energy that makes that signal happen. But the magic is really there at the synapse. That's where things really happen. That's where who you are manifests in the brain. So sometimes we think of the computer as the brain like a computer. It's much more like a network than like a computer. A computer is very synchronized. Everything works exactly according to a plan, and there's one set of instructions. And a network is where we have a whole bunch of different computers, all of them doing a little bit different things, and together they create something interesting and greater than what you see in each of the parts. So the basal ganglia is where Parkinson's happens. Most of the time when we think about the brain, we think about, you think about your consciousness, your perception. Who you are is up there at the top in the front of the brain, the cerebral cortex. The back of the cerebral cortex is where we have things like the visual cortex, where images are processed. And then we've got the cerebellum down below, and I've got a remote control there next to it. There's a remote control there because the cerebellum doesn't really do much thinking. The cerebellum is where the brain figures out how to connect to muscles, how it tells the body to move, and it's the switching area for how, when you think about what you're going to do, when you think about moving your arms or your fingers, that it then turns into a coordinated muscle movement. But down there where I've got the lizard brain, the brain stem, that's the part of the brain that is evolutionarily the oldest. It's the simplest functions of the brain. But because the brain didn't just layer on this frontal cortex that has all these complex functions, it's always been connected to those earlier stages. So things that happen in the basal ganglia affect the basal ganglia, but they also affect the parts of your core brain, your early brain structures that moderate what happens in the frontal areas. So it's a very key part of the brain. And a way to think about how the basal ganglia plays a role in the things that we're aware that we're doing is like this. So let's say you're learning a new task. I always think about playing tennis. Your brain says, wow, what an interesting new task. And your frontal brain says, okay, I've got to twist my arm and swing the racket and everything like this. And often when you first start playing tennis, the ball goes right by you. You swing your racket, but you're a couple of seconds too late. And it's because there's all this work that's going on in your frontal cortex, in your forebrain, to think about how you're going to how you're going to make those movements. And it involves a lot of activity. So it's a lot of work for your brain. And your cerebral cortex says, wow, this is a lot of work. This is something I'm going to do regularly. I don't want to work so hard. The brain the brain wants to be lazy. So I don't want to work this hard. And so what the brain does is it needs to make these connections to the cerebellum. So because the cerebellum tells the muscles to work, but it doesn't think about things. So the basal ganglia is where we create this switching there, where your brain first says, just to hit that tennis ball, it says, rotate the wrist and bend the elbow and swing the shoulder and rotate the torso and take a step up in your frontal brain. And then you get to the point where your frontal brain says, hit the ball. And your basal ganglia is the one that has simplified that hit the ball set of instructions to turn that into a coordinated set of muscle movements that all come out of this base structure. So what is Parkinson's disease and how does Parkinson's play into it? Parkinson's disease is a progressive brain disorder. It has a lot of symptoms, and people talk about how that it's not just a motor disease, and that's true. But as Parkinson's manifests, we recognize it by its effect on movement. That's the thing that is consistent across all different experiences of Parkinson's disease, is that it has an effect on movement. That doesn't discount the effect on other systems. It doesn't mean that Parkinson's related mood disorders or Parkinson's related sleep disturbance are not important. It just means that we diagnose it by the effect Parkinson's has on movement. So when we think about what all encompasses Parkinson's disease, we think about the motor disease with stiffness and slowing. There can be tremor. Not everybody has it. Changes in things we try to do, walking, freezing, changes in writing, changes in speech. And it's a non-motor disease. We see changes in mood, changes in how people think, the thought patterns, changes in sleep. There can be anosmia, loss of the sense of smell, lightheadedness, constipation, all these things that we think that are non-motor. So it has a broad array of effects. And when we manage Parkinson's disease, when your neurologist manages Parkinson's disease, they'll talk about medications, we'll talk about therapy, physical therapy, occupational therapy, speech therapy for people who need it. There's the self-care elements, exercise, your own practice to do the activities that are recommended by a therapist. We have surgical treatments like DBS and the pump for levodopa, for carbidopa levodopa called Duopa. There's social support programs. We wanted to have a community that can support Parkinson's activities of daily living, the things that we want to do. There are dietary interventions. But medications are something that really almost everybody with Parkinson's is eventually going to need. And the first line treatments for Parkinson's are to replace dopamine. So dopamine is a chemical for signaling in the brain that is when we think about the brain networks that are affected by Parkinson's disease, we think about the brain networks that use the neurotransmitter dopamine for communication. So we don't have drugs for every symptom. And just because there is a drug for a particular symptom, it doesn't mean you should take it. And some aspects of Parkinson's disease are better treated by non-medical interventions like exercise or diet. But replacement of dopamine is a key component. So what is dopamine? Dopamine is it's a neurotransmitter, which means it's a chemical that transmits a signal between brain cells. And very simply, the way dopamine works is there's a neuron that wants to send a message, and then the dopamine goes across the synapse, the little gap between the two neurons, and the neuron that receives the message receives that message because it gets that dopamine, because that dopamine crosses that gap, that synapse. So in people with Parkinson's disease, they're not synthesizing dopamine as effectively. And so that dopamine, that little packet of dopamine may cross the synapse, but there may not be enough of it that the receiving neurons knows that it received the message. It may be too little of that. So when we replace dopamine, sometimes it restores that normal function, but sometimes we just flood that gap. We make sure that there's just a lot of dopamine there, and that the signaling pathway is activated by a medication.
Dopamine was identified first in 1959 by Arvid Carlsson, and it was linked to Parkinson's disease by Ole Hornikewicz in 1960. So Hornikewicz was a neuropathologist, and he figured out that there was this substance called dopamine. They weren't sure what it did, but it was really low in people with Parkinson's disease. That kind of graph, when we, modern scientists don't see this kind of graph in a human study lately. We're much more subtle. This is a huge difference to see that the bottom of the error bar in the normal is that far above the top of the error bar in the Parkinson's. And this is in the caudate nucleus and the putamen, two parts of the brain that are affected by Parkinson's. Arvid Carlsson discovered that levodopa was part of the dopamine synthesis pathway. He figured out what the dopamine synapse looked like. He figured out that dopamine was a neurotransmitter, something that was not known before. They knew dopamine existed, but they didn't know what it did in the brain. So Carlsson figured out that it was synthesized from levodopa and was a neurotransmitter in a special kind of dopaminergic neuron, a neuron that uses dopamine. So when we diagnose Parkinson's, we can find and this is after people die, we can look at their brains, and in a healthy patient, there's an abundance of these dark pigmented cells in a healthy patient, and those are our dopamine neurons. And in a person with Parkinson's disease, those cells are gone, and they're really they can be seen with the naked eye, and then in staining, we can see that those neurons are gone. This is a stain called tyrosine hydroxylase. In the healthy patient, those cells are quite abundant. And in a patient with Parkinson's disease, there are far fewer of those cells. But then we note, when we look closely, there's something called a Lewy body, which is a little lump of protein that you can find in a patient in the neurons of a patient with Parkinson's. So the science advanced as Anne Grable in the 1970s was figuring out the pathways, the connections in the basal ganglia. In 1982, we had the discovery of MPTP, and Malin DeLong then figured out that using this toxin that we could surgically fix the deficit in dopamine producing. We could surgically fix the fact that when Parkinson's disease removed the benefit of the substantia nigra, that we could inhibit the function of the subthalamic nucleus, and that would re-normalize the signaling and give us a lot better motor control. And that was what led to the discovery of deep brain stimulation. So we went from the discovery of dopamine to where we understood that Parkinson's was a disease that could be controlled by medication that you take orally, a pill that you take every day, to the understanding of the signaling pathways in the brain, and that gave us DBS. DBS was a big improvement for patients who were having motor fluctuations where sometimes their medication worked and sometimes it didn't. DBS gave this huge improvement, and that was approved by the FDA in 2001. So right now, when we look at what's going on scientifically, there's good reason to think that the next breakthrough is coming. Parkinson's is a disease that's linked to alpha-synuclein. Could alpha-synuclein help us to this protein alpha-synuclein, which is the principal component of the Lewy bodies that we saw in the pathology slide? Could Parkinson's disease give us greater insight into, could our understanding of alpha-synuclein give us greater insight into the development of Parkinson's disease? Maybe, we hope so. But we're hoping that we had that great breakthrough from understanding that dopamine, that Parkinson's disease was a disease of dopamine. We then had a great breakthrough from understanding that Parkinson's disease was a disease of pathological signaling. And now we're hoping that one of many trials of drugs looking at the underlying biochemistry of Parkinson's disease are going to give us new insight into a treatment that could maybe stop Parkinson's disease from ever manifesting. So when you look at dopamine and we think about taking Sinemet, what we're really doing is addressing a cellular symptom of the disease. It's symptom management, because the symptom is your dopamine neurons are not producing enough dopamine, and we're giving them the benefit of a precursor chemical to help them produce more dopamine. When we look at DBS, we're looking at a organ system symptom that, because of this problem with dopamine in the substantia nigra, that the subthalamic nucleus is inhibiting the normal performance of a motor nucleus in the brain, and that by electrically slowing down the function of that subthalamic nucleus, that we can restore normal motor control. But now science is advancing in a way that is giving us new insight into the biochemical processes that lead to Parkinson's disease. So it's a very promising time in research, and we hope that this is going to result in something meaningful that we can bring into patient care. So let's now get back to COVID. What about COVID? How is this connected? So as we understand the layers of disease better, we understand its causes. So the layers of disease: phenotype, slow movement, right? We understand that Parkinson's disease is a disease of slow movement. We understand the anatomy of the disease. Well, it happens in the brain. In 1960, they discovered the connection with the caudate nucleus and the putamen. Then we discover it's a disease of networks with the research by Anne Grable and Malin DeLong. Then we understand it's a disease of cells, that the cells are having those Lewy bodies and need to produce that dopamine in order to communicate. Now we're understanding cell structures, that the microtubule deficits and the synapse involvement in Parkinson's, and then then we're digging down to the biochemistry. How does this protein alpha-synuclein get involved? So why do we worry about Parkinson's after viral infection? So we have to go all the way back to 1918. And in 1918, scientists discovered that there were neurological problems that came after the virus. So there were initial concerns from a paper in 1918 called where they discovered a post-viral illness that was von Eckmo's encephalitis, or encephalitis lethargica, that was discovered in 1918. And then in the 1950s and 1960s, they discovered that there was a link between people who had this encephalitis and people who later developed Parkinson's disease. So this graph with the arrows shows this peak in 1940, 1950 of people who had Parkinson's disease. But what they linked it to, what they've discovered was that the people who developed Parkinson's developed it at the same rate as they developed this von Eckmo's encephalitis by age group. So if in 1920, it was a 25-year-old who developed this von Eckmo's encephalitis, in 1940, it was a 45-year-old. So they were able to match up these age cohorts and say the people most affected by the encephalitis lethargica then became the people most likely to develop Parkinson's disease. So,
and I'm sorry, just to make the connection, and the people who got the encephalitis, they were more prone to get it if they had the 1918 flu. Is that?
So that wasn't proven yet.
Okay.
This is just this statistical connection that the age cohorts that developed the encephalitis then became the age cohorts that developed Parkinson's. So it was subsequent to that they discovered that viral Parkinson's was Parkinson's. And so the Melvin Yarr and his colleague Roger DeVossen discovered that there was this connection. They looked directly at patients and found that patients who had that had encephalitis then had Parkinson's disease. In 1983, in an animal model, they discovered that the 1918 flu did actually affect the brain, and that there was this risk that it was caused by that. And then in 2009, a group that included my colleague, who I've worked with, Richard Schmidt, showed that the descendants of the 1918 flu could then produce Parkinson's-like pathology. So it took until 2009 to close the loop. There's this speculation for decades that the 1918 flu was actually the cause of this wave of Parkinson's disease, and it really wasn't effectively demonstrated until 2009. So,
and so it became actually more, that was more genetic, like people that got the 1918 flu, their children were more prone to getting Parkinson's?
No.
Oh,
no,
okay. I thought you said something about their, okay.
No. So this was all retrospective. All the survivors of the 1918 flu were gone by, or almost everyone was gone by 2009. But Richard Sweeney in models using human cells in a petri dish discovered that you could get this Parkinson-like pathology, and that those patients in the 1940s and 50s who developed Parkinson's disease probably had gotten it as a result of the 1918 flu pandemic.
Okay.
So as we did this standard experiment emerged where you'd have an animal that was infected with either the virus or saline, and then those groups were split in two, and a toxin that causes Parkinson's disease would be given to one group and saline would be given to the other. That gives us 4 groups: virus and toxin, saline and saline, and each of the crossovers. And then we look at a bunch of different things, and this pattern was seen where, so this is a study of H1N1 influenza virus, the actual 1918 virus. And that it resulted in a modest increase in susceptibility to a toxin that's linked to Parkinson's disease. So the study becomes, it effectively becomes, does having had the disease activate your brain's immune system? The brain has a different immune system than the rest of the body that's centers around a group of cells called microglia. And the question is, does infection with a virus cause activation of the brain's immune system, which makes the patient more susceptible to a toxin, such as you might find from pollution? So is it opening the blood-brain barrier? So we have a pattern here, and that is that the least affected is the control, then the virus, then the toxin plus the virus. So having the virus plus the toxin gives you the largest decrease in dopamine neurons, the highest increase in activated microglia. So we've done this. This is a standard thing to do. People have done this with many different viruses, from herpes, Epstein-Barr, influenza A, the chickenpox virus, polio, HIV, all down the list. So why does this result in Parkinson's? Why do we think of this as a Parkinson's thing and not just are these Parkinson's scientists looking for Parkinson's, or are they Parkinson, or is there a reason that it would be Parkinson's? And the reason that it's Parkinson's is the basal ganglia, the part of the brain that's affected by Parkinson's, uses a lot of energy. It has to do this regulatory work across the brain, from the cortex, where your personality lives, to the cerebellum, where your muscle control lives. All of those things are really coordinated in this small region of the brain. And so it uses a lot of energy. It uses a lot of nutrients. It has a lot of blood flow through it. And because it's got all these things, it also has a very strong immune presence. These microglia are very present in the basal ganglia. So whatever is happening, you've got a lot of the blood flow and a lot of those nutrients and whatever the blood is carrying coming into that area, and a lot of this immune strength there. So early on in the pandemic, my team started thinking, what are we going to see after the pandemic? And I like this person points out that what we thought about that was scary about polio was not acute polio. It wasn't acute infection by the polio virus. It was long polio. And now we're looking at long COVID. What is long COVID? So I put together a team and we launched this experience. Before the pandemic, we were looking at doing a Parkinson's model in an animal. And so I put together a group. We had this MPTP, this toxin, and we had a vet who was trained in its use. Then COVID-19 hits. We received significant funding to support our research effort, and that resulted in $15 million coming to my medical school. And I was assigned the role of overseeing the spend of $15 million in 6 months. It was a little bit like Brewster's Millions, where we had to think about how do we make work happen in that time. One of the things I did was I renovated a BSL-3 lab, the kind of laboratory where you wear spacesuits and you can do research with highly virulent viruses. And we thought, we're not sure how long COVID is going to be, how long the pandemic will last. We foolishly thought that maybe we'd be able to stop it. And so I needed to have research that would carry us into the future. And then we started seeing reports of neurological symptoms, that people were having. Brain fog was the first to be reported, but I've talked to people who said that they've seen patients who manifest Parkinson's disease after COVID, and they were wondering if there's a connection. So I had all these pieces, and then I was friends with Rich Schmidt, who had done some of the seminal work in this virus to Parkinson's research space. So we have our beautiful new BSL-3 lab. I've got my team here. They are conducting the experiment. We have some animals that were infected with the virus, and then they were either given saline or this MPTP chemical that causes Parkinson's. So our first finding was that COVID-19 is dose dependent. And I just say this because it's interesting. This was an interesting finding for me. It's not related to Parkinson's, but our team found that COVID-19 was more sensitive to the dose of the virus that you get an infection than many other viruses. So we had to figure out what dose we were going to use, and we used this 4 × 10⁻³ units of virus. We found that if we used 1 × 10⁻³, all our mice survived. If we used 1 × 10⁴, all of our mice died. We had 75% of our mice died. So we used this 4 × 10⁻³ as our dose. What's important about this is it says that anything you can do to reduce your exposure to the virus is going to be a benefit. So it doesn't have to be the world's best mask. You don't have to stop all exposure in order to protect yourself. Anything you do that reduces it, a paper mask, a cloth mask, staying 7 feet away from somebody instead of 6 feet. All these things can help make it a little bit better and reduce your infectious dose a little bit so that if you get it, you'll have a much more benign course. And this may explain why so many people have relatively benign experiences. It may not be that something they did kept them, it may not be that something about their immune system protected them. It may be that they just were just that far enough away that they've got that very low infectious dose. Our second finding was that animals that were infected with COVID lost their dopamine neurons. So we found this reduction in dopamine neurons in the animals in the bar with a green circle on it. And for comparison, I've got that H1N1 experiment there on the right. And you can see that in the gray bars showing the COVID experiment, there are much bigger differences than in those colorful bars on the right, showing the loss of dopamine neurons in the H1N1 experiment. So the second thing we found was that COVID activates the brain's immune system. So we saw the same trend that it went the control, and then the virus only, and then the toxin only, and then the virus plus the toxin, but a much larger difference between the two groups. So there was greater synergy. Virus plus toxin yielded much more activated microglia than virus plus toxin did in the H1N1 and magnesium experiment. We found that the experimental model looks like Parkinson's disease. So when we look at our experiment on the left, these are slices of the animals, and you can see that when they were untreated, you can see those dark pigmented cells. So these are actually stained, but you can see those dopamine cells. And in the bottom slide, where it's SARS plus MPTP, you can't. And then remember, then in the H1N1 experiment, you can also see the loss of those pigmented cells. And just as a reminder, there's the human Parkinson's, where you can see that those pigmented cells are gone in the Parkinson's disease patients too. So we found that this experiment produced brains that they produced a pathology in the brain that looked a lot like Parkinson's. This study is currently in review. We're hoping to have the final draft sent back to reviewers sometime this week. I think we're going to send it in this weekend, and then it'll be in a refereed publication. We're very optimistic that we'll have some good outcome from that. This model, this approach that we've done, has been proven to show an increase in Parkinson's disease in a prior study. So this is a study of influenza that was done in Denmark. And what they found was that influenza infection was linked to an increased risk of Parkinson's more than 10 years after infection. And that's what they found. Now, the reason why this is important is that people talk about Parkinson's pathology starting 10 years before patients manifest. So you may have seen in other places where they talk about when we would need to intervene to cure Parkinson's. We talk about how early or prodromal stage patients are important to reach. And this study found that people who had influenza had a significantly higher rate of Parkinson's disease, about 75% higher rate of Parkinson's disease, starting 10 years after their influenza. No difference among people who had Parkinson's less than 10 years. So what you see is that it looks like this supports the idea that influenza can start the series of events that lead to the development of Parkinson's disease in a clinical population. And we know that the influenza virus is linked to similar pathology to what we saw with COVID. So we think that that means that the PD rate at 10 years after infection could be increased in a similar way, 75%, roughly double. So it's the same thing. So in conclusion, I think Parkinson's is going to increase over the next decade. We know it's going to increase because up until 2019, there were worldwide survival gains. People were living longer. But it's possible that it will further increase in people who've been infected with COVID-19. So Parkinson's disease only, is about 1.25% of people over 55. If that number doubles, it's not like everybody who's had COVID is going to get Parkinson's disease, but it will be an increase in the rate of Parkinson's disease, and it'll increase the burden of Parkinson's disease on health systems. So we need to, it's important that we develop more and more support for patients. We're going to need to think about how over the next 10 years, we're going to get ready for there to be more Parkinson's. Can we take these breakthroughs in the clinical science and see if we can slow down Parkinson's disease, reduce the burden of Parkinson's disease with new treatment? Can we engage with people to help them recognize when they have early Parkinson's disease, if there's a drug available that could slow the manifest, which we hope to have. Engaging with groups like the Davis-Phinney Foundation to do what you can to build your healthcare team and stay healthy, even though you've got some Parkinson's pathology happening and hopefully being slowed by medication and by self-care. So we need to think about how we're going to scale up our efforts to meet this potential burden. And lastly, I really hope that my research is wrong. Hopefully we will find that there isn't an increase in Parkinson's disease, but we need to be ready for it in case this is right. So thanks for listening. This is one of my favorite Banksy pictures.